सब्सक्राइब करें हमारा यूट्यूब चैनल, बेल आइकन को क्लिक करना ना भूलें, जिससे आगे आने वाले सभी यूट्यूब अपडेट्स, वीडियोस, नोटिफिकेशंस आपको मिलते रहें।
Namaskar. Welcome to the Parishkar College of Global Excellence, Autonomous. Today we will discuss the model paper for biology. In this one, first we will discuss the general instruction. You have to follow these general instruction while attempting the paper. So first one is all questions are compulsory, and in this one the question paper has 5 sections. In this one, A, B, C, D and E, and in these sections there are 33 questions. So out of these sections, first questions contain the 16 questions from 1 to 16, and in this one, each question carry 1 marks, and in the second section there are 5 questions 17 to 21, 2 marks each. In the third section, C section, there are 7 questions 22 to 28, 3 marks each, and in the fourth section there are 2 questions and 29 and 30, and in the fifth section there are 3 questions 31 and 33. So each that question is carrying the 5 marks. So there is no in the question there is no overall internal choice, but in the some questions you may get the internal choice, so you are able to solve the any questions from that internal choice. So that are there, that is available in the some questions. Out of these one, you have to make a good diagram. Whenever there is a requirement of a diagram, then you have to make a good diagram and label it properly. So first we will discuss the section A, and in that section all the questions are objective type. So look at this one. Remnants of nucellus are persistent during the seed development in. That means you have to first look at this one. That is, they are asking about the nucellus, and in which one the nucellus is persistent. So what is the answer? So first thing is that what is nucellus? That means if we discuss about the ovule, and in that ovule we found that nucellus is present, and that nucellus is the structure in which the female megaspore is developed, and that megaspore later on converted into a female gametophyte. So this nucellus is developing in the ovule, and later on during the development of embryo and during the development of the gametophyte, this nucellus is consumed. But there are some plants, there are some examples in which the nucellus is remaining, and that nucellus is present later on in the embryo also. So which one is the example in which the nucellus is remaining? And that nucellus is remaining in the black paper. So the right answer is D, black paper. In this one, nucellus is remaining, and that nucellus is known as perisperm. So whatever be the nucellus is remaining in the seed also, that nucellus is known as perisperm. So that is the first question. Now the second one is the wall layer of microsporangium, which nourishes the pollen grain. Look at this one. Microsporangium, that means we are talking about the male development in the flower. In microsporangium, microspores are developing, and that microspore is later on converted into a pollen grain, and that pollen grain is male gametophyte, and that male gametophyte is developing in situ. So which layer of the microsporangium which provide the nourishment to the developing male gametophyte? We can say that pollen grain is also known as male gametophyte. So look at this one. Which one is the layer which is providing nourishment to the pollen grain? So that answer is tapetum, because that is the innermost layer, and that innermost layer is surrounded by this microspore mother cells, and in that one microspore mother cells the meiosis takes place in these microspore mother cells, and these after meiosis they form the microspores. So whatever be the layer is present surrounding these microspore mother cells, that layer is known as tapetum. So that tapetum is taking the nutrition from middle layer, and that tapetum is supplying that food material to the microspore mother cell, and microspore mother cell did the meiosis, and after meiosis they are converting into the microspores, and these microspores are later on developed into the male gametophyte, and that male gametophyte is known as pollen grain. So that whole structure is known as microsporangium, and that microsporangium is present in the anther. So that is regarding to the development of male gametophyte. Next question is, now look at this one. A short piece of DNA having 20 base pairs. So there is a short segment of DNA that contains the 20 base pairs was analyzed to find the number of nucleotides. So we know that DNA is made up of nucleotides, and that nucleotide is present in a series, in a sequence. So in this one, that pair is 20 base pair. There are two strands, one strand and second strand. Nucleotide base in each of the polynucleotide is sum of the result. So out of these 20 base pairs, there are some conclusions in adenine, cytosine, guanine, thymine. In first strand, they found that adenine is 4, cytosine is 4. And in the second strand is they found that cytosine is in the form of nucleotide 5. So they are asking that how many nucleotide containing adenine, that means in second strand what is the number of these adenine nucleotides in the second strand. So we have to calculate that number. What is that number? That okay, this one is the number of the adenine nucleotide in the second strand. So we have to name these nucleotides. So which one is the number of these adenine nucleotide in the second strand? So look at how to solve this one. So right answer is 7. Look at this one. First one is adenine, second one is cytosine, third one is guanine, and fourth one is thymine. In this one, in the first strand, they show the number adenine is 4, cytosine is 4, and in the second one, the second strand, they show the number of cytosine. So how we have to solve this one? This one is very interesting. Look at this one. In the DNA, adenine always bind with the thymine. In the DNA, cytosine always bind with the guanine. Okay, so that they are by triple bond. They are bound with each other in this way, Chargaff rule. So if they are joining with each other by adenine and thymine, cytosine and guanine, so how we have to calculate that number that adenine is second? Look at this one. So in this first strand there are 20 numbers. Total number of nucleotide is 20. Out of these 20 nucleotide, 4 is adenine, 4 is cytosine. So in the second strand there is also the nucleotide number is same. So look at this one. If cytosine is 4, that means guanine is 4. If in the adenine they are 4 and 4. If cytosine is 5 in the second strand, that means out of these 5 cytosine, they bound with the guanine. So there are 5 guanine also. Now look at this one. Out of these 20, if we found that 5 is the number, and if 5 is cytosine, then they bound with the guanine. So what is the number of adenine? 5 and 4, and 4 plus 5. So that 5 number, and when they calculate with the cytosine, then adenine is also bind with the thymine, and we calculate that this number is increased to the 13. So remaining number is left with the 7 number, and that 7 number is of adenine. So right answer is adenine. Now next one is, in a certain species of insects, some have 13 chromosomes and the other have 14 chromosomes. So there are insect species, and in that insect species they have the 13 and 14 chromosomes. So in that one we know that what is this one is male or female. So there are 13 chromosomes and there are 14 chromosomes. So out of these 13, which one is male and which one is female? So that is, in the insects there is a development of sex determination, and that sex determination is based on the number of chromosomes. So in that one, X0 condition, and X0 condition is converted into the males. So because of that reason, there are, if the less number of chromosome, so that was 13 chromosome is the male, and which one is that containing the 14 chromosome, that is the female. So because of the number of chromosomes and number based sex determination in the insects, so less number of chromosome male and that more number of chromosome, that is the female. So right answer is A. Now next one is, at a particular locus, now the frequency of allele A is 0.8 and that of allele A, small a, is 0.2. What would be the frequency of heterozygotes in a random mating population equilibrium? Now look at this one. They are asking the heterozygote frequency of heterozygotes. So in this one they are given us the choice, and in that one Hardy-Weinberg genetics is based on three parameters. First one is classical genetics, second one is molecular genetics, and third one is population genetics. Third one is population genetics. So in the classical genetics, we are studying the transmission of the character from one generation. In the molecular genetics, we are going to the molecular level, that DNA is, RNA is, and how that central dogma and these genes are expressed in the form of character. Now next one is, in a population, the species constitute a population, and in that population how these genes are behaving. For this one, Hardy-Weinberg given a law, and in that one, look at this one, P plus Q is equal to 1 at a equilibrium, their amount is 1. And after that one, we calculate that P square plus 2PQ plus Q square is equal to 1. Now in this one they have given us the heterozygous condition. That means heterozygous condition is A and capital A and small a. So in this one we have put down the direct number of this one, and when we put down direct number of this frequency of this one, then we found that the number is 0.302. Okay, so in Hardy-Weinberg law, they are showing us the what is the frequency of population and the frequency of the genes in the population. So that frequency is calculated by this formula, and in this formula they are asking about, they gave us the heterozygous condition frequency, that is the capital A, and that that recessive allele, and in that recessive allele the amount is 1. So we can calculate by putting this heterozygous condition the number of frequencies in each place, and we calculate the number is 0.3 to the answer. Now next one is the variations caused due to mutations are. So what is the option in this one? Now look at this one. There is a genetic material, and in that genetic material all the informations are stored. In our genetic material all the informations are stored, and there is a two way by these informations can be changed by two ways, and that way are one is natural process, and that natural process is known as recombination, and second process that second process is known as mutation. So we can change the information which is present in the genetic material by two ways, that is recombination and by mutation. So that recombination is the natural process, and in that natural process that happens naturally during the formation of gametes in the meiosis. So recombination takes place naturally, but there are some mechanisms by which there are sudden changes in the genetic material, and by which we are able to change the information which is present in the genetic material, and they are heritable. So because of that reason, that whatever be the changes created due to mutations in the genetic material, that is known as variation. So what are these variations? How these variations are they are permanent in the cell? Are they permanent in the organism? So look at this one. They are random and directionless. They are random and directional. Random and small, random, small and directional. So which one is the option about the variation which is caused due to the mutation? So that option is first one is correct, random and directionless, because by mutation you can create any change in the genetic material. So they are directionless. They are, if they are found by naturally happens, they are happens in the nature, then they are random in any nucleotide, happens in any nucleotide, and direction, there is no direction to the that this mutation or this change can happen in this particular nucleotide. So that is the random and directionless. Now next one is, what is the smallest part of DNA molecule that can be changed by point mutation? First one is mutation. That means there is a change, heritable changes in the genetic material. And if we are talking about point, so point means that particular place, and in the DNA structure the particular place is the nucleotide. That particular place in the DNA, because we know that the DNA is made up of nucleotide, and that nucleotide is made up of nitrogen base, sugar and phosphate. So that particular nucleotide, if you are changing any particular place, that means you are changing the nucleotide of a DNA. And when you are point mutation by the, that means you are creating the changes, and that changes in the nucleotide. So point mutation is always created change, and that change is in the nucleotide place. Next one is, that is the pedigree analysis, and in that pedigree analysis very much simple one. So look at this one. They are if they are breeding two individual, and in that two individual they are asking the what should be the genotype of indicated member, and that is the indicated member, and this one is the indication, and this indication is if the genotype is expressed, and that genotype is expressed in the recessive condition, that recessive condition always expressed in the homozygous condition. So in that pedigree analysis, simple one is that very much interesting. Pedigree analysis, there are huge number of questions in the pedigree analysis, but in this one there is simple one, that homozygous recessive character is expressed, and because homozygous recessive is expressed, so that is because of homozygous condition. So answer is D. Next one is, a patient was advised to have a kidney transplant to suppress the immune reaction the doctor would administer him. So what is that medicine which is given by this one? So this one is a factual questions. In this one there is not that much of Explanation. So right answer is C. Cyclosporin A produced from Trichoderma polysporum. So that is the medicine, and medicine is obtained from this one. So that is the simple answer. Now next one is the identify the activity of endonuclease and exonuclease in a given image. So look at this one. They are very much important enzyme, endo, and the name is exo. So there are two names, endo and exo. Endo means they are cutting from in between, and exo means they are cutting from the end of the nucleotide. So they are very, very important enzymes in our DNA replication or in the central dogma mechanism, and they are very useful enzymes that in the biotechnology also. So in recombinant DNA technology, they are very much important enzymes. So one important thing is endonuclease cut the DNA from inside, and the exonuclease cut DNA from the one end, and in that strand the DNA is made up made up of two strands, and when that made up of two strands, that means there are one strand and that is second strand, and that both strands are anti-parallel. So one strand is 3' to 5', and another strand is 5' to 3'. So out of these, one exonuclease can cut the DNA from in the middle of the strand, while the exonuclease always cut the DNA from one strand. So look at the answer. Which one is the answer, that how they are cutting? So look at why that first one is incorrect. That same concept is following the another option. So look at this one. This one is 3' and 5'. If this endonuclease first cut the DNA from this side, then this one is 3', then this one is 3', the against is 5', and after this, if it one is the 5', then this one is 3', and next one is if cutting, then 5', then 3'. One option. But what is the problem with the exonuclease? Exonuclease always cut with the end. So if this one is the 3', then this one is the 5'. Next one is this one is the 3', and that this one is the 5'. And so look at this one. That concept is following for every strand. So right answer is D. Why that answer is, that is 5'. Now this one is 3'. They always cut the DNA from one end where they found the particular group, and when they found that particular group, so because of this reason they are cutting the DNA from this end. So this one is the 5', this one is the 3'. So look at this one. If this is the 5' end, then open end is 3' end, and the cutting DNA, that is 5', 3', and that same concept is applying to everywhere. So right answer is D. Because of the presence of 3' OH group, they are cutting DNA on a particular place. So because of that reason, that is the right answer. Now next one is the main objective of production of pest resistant genetically modified crops. So two things, pest resistant and genetically modified crops are developing because of biotechnological development and recombinant DNA technology. And in that recombinant DNA technology, we are inserting a foreign gene into another crop, which is very much useful for that crop, either for the crop production, either for the resistance to the pest, whichever organisms which are affecting to that particular crop, either the insect or anyone else. So why we are inserting that pest resistant gene into a particular organism? So what is the main objective? What is the main objective of this one? Why we are inserting that particular gene? What is the problem of Punjab? In Punjab, there are huge pesticides, they are growing the crops on the basis of pesticide. There are huge number of pesticide, and that pesticides are keep on accumulating in the food chain. We know that there is a ecological concept, and in that ecological concept there are organisms are organized hierarchically according to food habits, and in that food habit, if they are keep on eating, and we are applying pesticides in the primary level, that is the production level, and if your primary producer is doing these things, that means the plants are keep on accumulating that pesticides, and that pesticides is keep on accumulating in the food chain, and that ultimately goes to the that ultimate plant that animal, to the which one is the in a highest level of the food chain. So because of that reason, we are developing the pest resistant varieties, so that we have to protect this food chain to by inserting these pesticides in this one. So right answer is reduce the pesticide accumulation in the food chain. That is our ultimate goal. So because to save these animals or these whichever human being is depending on the food chain. Okay, so right answer is B. Next one is observe the content 1, 2, 3 and 4 of soil sample. There are 4 soil samples, A, B and C. So these are the soil samples. In these soil samples, soil sample 1, soil sample 2, soil sample 3. In these 3 soil samples, there are different type of contents, and in that content, first one is lignin content, second one is chitin content, third one is indicating nitrogen content, or fourth one is indicating the sugar content. There are 3 soil samples. In each soil sample, there are different concentration of biomolecules, and in that biomolecules, if the concentration is of different for the different biomolecules, so which soil sample exhibit faster the decomposition, if the temperature and soil moisture of the sample are identical. In all the 3 samples, they have the identical temperature and moisture conditions. So which soil sample exhibit the faster decomposition? So that is by the soil sample D. Now look at this one. If we compare in this one, we have to compare that samples, that which sample has that have the first one is lignin content, second one is chitin content, third one indicate the nitrogen content, and fourth one indicate the sugar content. So look at this soil sample. In that soil sample, lignin is very much less. After that one, chitin is very much less, because we know that this lignin and chitin, they are not that much degradable faster. So if they have the more amount in a soil sample, that degrade the very much slow in slow speed. So if we go through the content, average content, what is the amount of that particular hardening content, which is very much less in speed to for the degradation, in that we found that soil sample C, where that amount is very much less, and because of that reason, they are showing the faster rate of degradation. So right answer is soil sample C. Next one is, there are questions 13 to 16 number questions, and in that in these questions you have to write the assertion reasoning type of questions. So in assertion reasoning type, you have to follow these guidelines, that okay, what is the answer of this one. So we will go through the 13 questions. Assertion: primary endosperm nucleus is diploid. So primary endosperm nucleus, it is the product of double fertilization. So look at this one. First one, assertion: primary endosperm. Endosperm is a nutritious tissue. Endosperm is a nutritious tissue and that provides the nutrition to the developing embryo. Second one, endosperm is diploid. How it is possible? Because in angiosperm, endosperm developed after double fertilization. It is correct. And in this one, look at the endosperm is developing after fertilization by the fusion of polar nuclei and one of the gametic nuclei. So because of this reason, that polar nuclei is diploid and that gamete nuclei is haploid, so that endosperm is triploid in nature, that is not diploid. So the right answer is A is false but R is true. So because this primary endosperm is a triploid in nature. Okay. Next one is assertion ribosomal RNA is synthesized in the nucleus of the cell. Ribosomal RNA is synthesized in the nucleus of a region. It is translated with the enzyme RNA polymerase. Look at RNA polymerase third. It is sure that okay, this one is ribosomal RNA is synthesized in the nucleolar organizing region. It is synthesized in the nucleus. But problem is that this is not synthesized with the RNA polymerase third, that is synthesized by the RNA polymerase second. So this one is the enzyme which is responsible for mRNA synthesis, not ribosomal RNA. So right answer is A is true but R is false. Next is assertion smoking can raise blood pressure and increase heart rate. Okay, by smoking, they both increase the blood pressure and increase heart rate. Okay, why? The reason is nicotine stimulate adrenal gland to release adrenaline and noradrenaline into the blood circulation, both of which raise blood pressure and increase heart rate. That is the conceptual question and they both are right. So both A and R are true and R is the correct explanation of A. Next one is assertion PCR, polymerase chain reaction, is a powerful technique to identify genetic disorders. Yes, this assertion is correct. PCR can detect mutation in low amounts of DNA. So the answer is A. Both A and assertion and reason are true and R is the correct explanation of the A. So that is the completion of section 1. In that section, we have discussed all the 16 objective questions. Now we have to go to the section B, and after this one there are subjective questions. So first one is explain the process of hormonal regulation of spermatogenesis. Now look at this. Either the plant or either the animal, whatever be the organism, there is a development of reproduction, and in this reproduction there are three processes: vegetative, asexual and sexual reproduction. If there is a vegetative reproduction, that means they are converting, the body part is separated and that body part is converted to a new individual. In asexual reproduction, that means there is a formation of a specialized structure and that specialized structure is converted into a new organism. But in the sexual reproduction, there is two processes, and these two processes are formation of gametes, and that formation of gametes is by meiosis, and after this one there is fusion of gametes. So because of that reason, the sexual reproduction is very much specialized, and that happens in both plant and animals. So if that happens in plant and animals both, that means there is a formation of gametes, and for the formation of gametes takes place in a specialized sex organs, and these sex organs are well developed in the animal kingdom. And in that we are talking about human being, and in human being that there is a male and female sex organ, and in that male sex organ there is a process of formation of sperm, and that is known as spermatogenesis, and that spermatogenesis is very much regulated by the hormones, and whatever be these two hormones, that follicle stimulating hormone and luteinizing hormone. They are very much important hormone. So how they are regulating? So first one is spermatogenesis start at the age of puberty. Okay, yes, that is there is two stages, vegetative growth and reproductive growth. So both in plant and animal. So whatever be the organism, whatever be the plant, they have to attain a certain stage for the sexual reproduction. So that is a stage in human that is known as puberty. After this one, when that attain the age of puberty, then this is due to significant increase in the gonadotropin releasing hormone. So these hormones are secreted, these two hormones, they are secreted by the hypothalamus region. Now these hormones are stimulating the glands to secrete two hormones, and these are luteinizing hormone and follicle stimulating hormone. They are very much important for development of these sperms by the spermatogenesis. Now look at LH, luteinizing hormone, act on the leading cells which are present in the testis. So in the testicular cells, so these leading cells, they stimulate these leading cells, and these leading cells secrete the hormones. These hormones are known as androgens. Now androgens in turn stimulate the process of spermatogenesis, and in that spermatogenesis they further act on the different cells, so they are able to develop mature sperm in the testis. Now that first one, this one is the first hormone, FSH, follicle stimulating hormone. That is the second hormone, and that second hormone act on the Sertoli cells, and these Sertoli cells are acting on the factors which are also acting on the spermatogenesis factors. So these are the two hormones which are very much important one. Now the next question is, diagram below show the sequence of amino acids in part of hemoglobin molecule. Hemoglobin molecule is present in our blood. That hemoglobin is very much useful for the carrying of the oxygen and circulation of oxygen in our blood. Now look at the central dogma. If the character is present and that character is expressed in a form of a character, that means that character must be present in the form of a information in the genetic material, and that character is expressed in the form of a protein. That hemoglobin is a protein. That hemoglobin is a protein. That means they are made up of amino acids, that there is a sequence of amino acids. So let us, that is the particular sequence of amino. Look at this one. This is the hemoglobin chain. This one is the mRNA which is carrying the information. This is the central dogma. In that central DNA information, that DNA information expressed in the form of mRNA, and that mRNA is ultimately expressed in the form of a protein, and that is a feature of a character. So look at this one. There are sequence of these nucleotides, and in that nucleotide sequence they form a code, and that code is triplet. Now look at this one. This one is the code, and that code is particular for particular amino acids. So look at this one. This one is the DNA. That is code expressed in the form of mRNA, then goes to the in the form of hemoglobin. Now look at what is the question. If the base, look at this one. This one is the base CTT. In this one was substituted with A. If one T, that middle T, is substituted by the A, then what is the result? Why that is replaced, and what is the effect of that T replaced by the A? That means thymine is replaced by the adenine. Now look at this one. Name the condition and the effect associated with the substitution. If in hemoglobin, that one code, in one code that thymine is replaced by the adenine, what is the result of this one? So look at this one. If in this one, this T, this T is replaced by the A, then that means that code is CTT is converted into the CAT. Now this CAT is for the valine amino acid. Okay, we know that this one is the this one is code for the particular amino acid, and if this one is for particular amino acid, that means CTT, that is now converted into the CAT, and this CAT is for the valine amino acid. Now what happens? Thus in the hemoglobin, that glutamic acid replace the glutamic acid by the valine, and because of that reason, that is one disease, and that disease is known as sickle cell anemia. Okay, so because of this replacement, the disease is generated, and that disease is known as sickle cell anemia, and because of that reason, the sickle cell anemia disease, you have to change that RBCs, which are change their shape, and because of changing their shape, they are not able to carry that much of oxygen. So that is the reason, and because of that reason, the disease name is sickle cell anemia. Next is, now there are vaccination. In that vaccination, we are providing a dose. We are providing a dose to a particular person to a fixed amount. So in this question, the graph given below indicates the administration of first. There are two doses, first dose and there is a second dose of vaccine to a particular person. So for that particular two dose, that particular person exhibit two different response. And why that these two different responses? We have to write about this one. So because the corresponding response of the body is indicated by X and Y. There is a vaccine. By this vaccine, we are providing two doses, first dose and second dose to that particular person, and by these two doses, we are calculating the different response to that response of that person to that particular dose. So what is that response? Why there is a two different response to that particular dose? Okay, so that is the question. In this one, that is the first dose, this one is the second dose, and during first dose there is a different response, there is a during second dose there is a different response. So for same vaccine, because of two dose of the vaccine, why there is a different response of that particular person, we have to explain and we have to give those reasons why that is happens. So we are writing. So look at this one. If that response is for particular immunity development, that is for particular antibodies development. If that is for antibodies development, then during the first response, what happens? During the first response, cell give response to that one. Cell have to identify that antigen, and when they identify that particular antigen, then they have to respond. That is the first one, because there was earlier that is not antigen, that no antigen is present in our body in that particular person. So when we give the first dose of that vaccine to that particular person, that organism or that particular person exhibit that cell recognize this one. After that recognition, they respond in the form of antibody. So because of that reason, there is a lower response. So look at this one. On administration of the first dose of the vaccine, the body shows a response of low intensity as the immune system comes in contact with the antigenic protein of the weakened or inactivated pathogen for the first time. So they are coming in the contact, they are identifying this one, they are forming the antibody. Now if we provide that same vaccine in the form of second dose, now look at this one. These cells are already in the contact of that particular antigen. Now they are able to recognize quickly, they are able to recognize that antigen, and when they are able to recognize that antigen quickly, then they are able to respond to that antigen quickly. So because of that reason, second response is very much faster and that very much higher one. So this response is known as primary immune response, that is secondary response. And when we provide the second dose to that one, the body elicit a highly intensified secondary response to that particular antigen. And that is happens that because of corona period, we are providing first vaccine, we are then second dose of that vaccine, then we gave that booster dose also. So that is the right one, that because of that reason, identification and the already presence of that antigen in the cell, they are able to respond quickly. So because of the memory of the first contact with the antigen, the secondary immune response is faster and stronger, leading to more effective pathogen elimination in comparison to the primary immune response. So because of that reason, in the vaccination, because our life is based on vaccination, after the birth of a child, we are start vaccinating that particular child, and there are huge number of vaccines, or they are giving after a particular interval. Okay, so because of this reason, they are giving the different doses to that particular person, to the particular antigen. So that is the very much important one, immunology. Now next one is the image below show the result of plating bacteria in chromogenic medium. That means that medium which show the change of a color. So that medium is known as chromogenic medium. That means there is a change in color of the medium after incorporating the gene of interest in plasmid. So there are two things here. First one is plasmid. What is plasmid? The plasmid is we have obtained the plasmid from bacterial cell, and that is the very much interesting research. Now that plasmid is extracellular DNA. That is extra DNA which is not that much required to that particular bacteria, but that is providing extra characteristics to the plasmid. But that plasmid is very much useful for us. That plasmid is providing us a vector. That means we are able to transfer genes from one cell to another cell. So if we incorporating that particular gene into that plasmid, and we are inserting that plasmid into the particular bacterial cell, that means some plates have blue colonies. We are identifying these characters based on the color changes, how that color is changes by by that particular bacteria. So there are three plates. In this one, there is a bacterial cell, there is a plasmid. In this plasmid, we have inserted that particular gene of interest. Now look at this one. We are inserting that particular gene at a particular place, and that particular place, how we have to identify that this one is inserted or not. We are inserting that particular gene in a particular place which is responsible for the character expression. And how that character is expressed, in this one we are identifying that look at, if an enzyme is synthesized, and when that enzyme is converting one product into another, that means there is a color change product. One product is converted into another, then at that time, if there is a change in color, we are able to identify that this gene is, our interested gene is inserted or not. So in first one, there is a insertion of gene. Look at the green gene. In second one, there is no insertion of the gene in the plasmid, and there in the third plate, there is no plasmid. Okay. So what is the conclusion? There is a bacterial cell. In that bacterial cell, plasmid is present, and in that plasmid, we are using that plasmid as a vector, and in that vector, we are inserting that particular gene. If we are inserting, then what is the effect of this one? So first one, identify the plates which are white, give a reason. Why these plates are blue, but why they are converted into a white one? Identify the plates which is blue, give a reason. So we have to identify these plates which are blue in color. So look at why. First one we have inserted, and we have inserted that gene into a place. That place is synthesizing the enzyme, and that enzyme is beta galactosidase. This one is the enzyme which is synthesizing that from a particular place. And what is the role of this one? The role enzyme is responsible for the blue color. That means this enzyme is converting a product into a particular product which is able to give the blue color. If that is giving the blue color, then we are able to identify that, okay, this one is the beta galactosidase enzyme. Now, if we are able to insert a foreign gene into this beta galactosidase enzyme, then what is the effect? The effect is that beta galactosidase enzyme is not able to form, and when this enzyme is not able to synthesize, then that blue color is not able to develop, because that product is not able to convert it into a product which is blue in color. So because of that reason, if you are inserting that gene into the beta galactosidase, so first one plate in the first, that first plate is able to insert, and that is not able to develop the blue color. Second plate, in the second plate there is no insertion of the foreign DNA into that beta galactosidase, so that is able to develop, and that is developing the blue color. In the third plate there is no plasmid. So if plasmid is not present, then there is no beta galactosidase gene. So we are developing that plasmid on the basis of insertion inactivation. We are developing that plasmid on a particular place of beta galactosidase gene, and in that one, by the insertion of inactivation, means we are inserting the foreign gene into that particular place. So because of that reason, that enzyme is not able to express. So that is the answer one. Now look at this one. That is the question from ecology. Biomass, that what is the organic matter is present at a particular place at a particular time, that is known as a biomass. Biomass of a standing crop of phytoplankton. There are hierarchically organized that organisms which are present in ecological system. They are hierarchically organized, and when they are hierarchically organized, how their food habit is present. So first one is phytoplankton, and phytoplankton has this one is the biomass of phytoplankton. Second step is there are zooplanktons, and zooplanktons have this much is the biomass. Third one is that is the consumers, and that consumers have this one is the biomass. And on the last one there are fishes, and that fishes has this one is the biomass. Okay, so look at this one. The biomass of standing crop of phytoplankton is this one, and that biomass is keep on increasing. That biomass is keep on increasing from first the producer level to the consumer level. So look at this one. Draw an ecological pyramid indicating the biomass at each stage, and also name the trophic level. Mention whether it is an upright or inverted pyramid. So that pyramid is in the ecology, there are different number of pyramid. There are biomass pyramid, there are that energy pyramid. So how these organisms are organized into a pyramid, and what is that shape of that pyramid. So look at this one. We have to make the pyramid. So look at it. This one is the phytoplankton, that these are the primary producer. Now in these primary producer, that biomass is this one. Now after this one there are zooplanktons, and they are primary consumers, and zooplankton, that these zooplanktons have this much is the biomass. Last one is there are secondary consumers, small fishes, and they have this biomass. After this one there are large fishes, and these large fishes have this much of the biomass. So because of this reason, they are small. after this one keep on increasing. So because of this reason, that is the inverted pyramid of biomass. So you have to make a diagram of this one and write the inverted pyramid of biomass. So biomass is keep on increasing in this ecosystem. Now next, that is the completion of section B. Section C, and in that section C, the first question, or that is 22nd question, is the figure given below. So 3 sperms, there are 3 sperms, A, B and C. First one, which one of the 3 sperms will gain entry into the ovum? So that is the ovum, and that is if that is the ovum, and out of these 3, which one enter into the in the ovum? And second is describe the associated changes by it on the P and Q. Look at this one. Q is the ovum, and in this P is the outermost boundary, or that zona pellucida of the ovum. So what is the effect of after entering into the ovum by the sperm? So first one, we have to know this one, which one is the able to enter into the ovum. So first one is A. So A is able to enter into the ovum. So after entering into the after entering of a sperm into the ovum, what is the changes? So first one is change effect on the zona pellucida. In the figure given, sperm A come into the contact with the zona pellucida layer of the ovum. It will induce changes in the membrane that will block the entry of the additional sperm B and C. Look at this one. Now first thing is that if there is a fertilization, that means sperm is able to enter into a ovum. That time after first entry, that is monospermy. If that sperm is, that first sperm has to stop that entry of another sperm into the ovum. So because of that reason, they have to develop the changes, and they have to develop the changes into these different layers of the structure of the ovum. So the outermost structure is zona pellucida, and after that outermost, that is the B and C layer, additional layers. So that change takes place in these additional chair, and these are able to stop that further sperm is coming into the contact with the ovum, and that further development of ovum, because that the secondary oocyte, and they have to develop the second meiosis also. So because of that reason, they are to stop the that entry of the another sperm into the ovum. So that ensure the only one sperm can fertilize the ovum. Now the secretion of the acrosome sperm A will help to enter into the cytoplasm. Yes, that is the mechanism by which that they enter into the zona, crossing the zona pellucida layer, that is outermost layer, the plasma membrane. This will reduce the completion of meiotic division, induce the completion of meiotic division of the secondary oocyte. So because in a human being, the entry of the sperm into the ovule start the second meiosis, and when that second meiosis is started, that is started in the second oocyte. So because of that, in the second polar body is able to separate from this one. So because of that reason, they are exhibiting this type of that first sperm is exhibiting this type of changes into the ovum for further development. The second meiotic division in the ovum being unequal, that result in the formation of second polar body and that haploid ovum. So that is the reason. Now the haploid nucleus of sperm A, that of the ovum, will fuse together. That is the obvious reason of the fertilization, that because of this reason fertilization process is taking place, and in that fertilization process, the whatever be the genetic material, that haploid genetic material is obtained from the sperm in the form of nucleus, enter into the ovum, and they are joined together, and that is a syngamy, and in that syngamy they are developed into a form of a zygote. Now next one is explain the phases in embryonic development from the morula stage till the establishment of pregnancy in human female. So look at this one. There is a development of zygote. That zygote is, after that zygote is converted into a multicellular embryo. When that is converted, that means the zygote is single cell, and when that single cell zygote is converted into a multicellular zygote, that is taking place in the uterus. So first one is that the embryo with 8 to 10 blastomeres is called morula, and that morula is later on developed into a particular embryo. The morula continues to divide and transform into the blastocyst. So first look at this one. Zygote is dividing, and that zygote is converted into a 16-celled stage, and all particular cells, each particular cell is known as blastomeres. That 16-celled stage is known as morula. When that enter into the uterus, at the uterus, that blastomeres further converted into a 2 layers, and when they are converted into 2 layers, that means the outer layers and inner layers, because that morula must have to attach with the uterus, wall of the uterus, because further development for the nutrition, for further development, that has to attach with the uterus wall. So because of that reason, that is differentiated into 2 layers, and that outer layer, which is known as trophoblast, that trophoblast is attached with the wall of that uterus. Now that uterus is growing, and that uterus is surrounding that this embryo, and when that is surrounding, then that is able to join this one. So here 2 differentiation: the outer layer attached with the uterus wall, and there is inner layer that is developed into a proper embryo. So that is the reason the trophoblast layer then get attached to the endometrium, and the inner mass get converted into the embryo. Now next one is, after attachment, there is a that is surrounded by the wall of this uterus, and that uterus is able to then form there is a formation of placenta, and that placenta is very much useful during the pregnancy and during the development, and that establishment of that embryo in the form of structure into the uterus, that is known as implantation, and that implantation is starting of the pregnancy. Now next one is pregnant women. Pregnant human female was advised to undergo MTP, medical termination of pregnancy. It was diagnosed that fetus she was carrying had developed from a zygote having 45 chromosomes. Human cell, diploid cell, have the 46 chromosome, but this zygote has a 45 chromosome. 1 chromosome is less. So what is that condition? What is that condition? And only having 1 X chromosome. So what is that condition is known as? What is this condition called, and how does it arise? Why was she advised to medical termination of pregnancy? So that condition, that is known as Turner syndrome. If they have the 1 X chromosome less, that condition is known as Turner syndrome. And we know that because of this reason, if 1 X chromosome, they are showing some abnormal features, and because of these abnormal features, we have to abort that fetus. Why? Because that baby is normal. So because of this reason, this exhibits some abnormal features, and because of these abnormal features, that is the MTP, medical termination of pregnancy, is authorized to that particular lady. So what are the reasons? That is the rudimentary ovaries. Ovary are very much reduced, poorly developed breasts, lack of other secondary sexual characters, they are absent, delay or no onset of menstruation cycle, or infertile. Because of that reason, doctor is allowing that particular lady for the MTP. So these are the reasons. Now next question is, the graph below show the 3 types of natural selection, because Darwin, now that is coming from the evolution, and in that one the graph below show the 3 types of natural selection. That means we studied in the evolution that there are organisms, and in that these organisms natural selection is acting, and when natural selection is acting, that means there is a selection of the particular population and species. So in this one there are three graphs. So shaded area marked with the arrows show the individuals in the population which are not selected. The dotted vertical line shows the statistical means. Look at this one. This one is a graph, and in that one number of individuals, these are the shaded region, and in these shaded regions they are selected. Okay, these are these population are not selected. Okay, rest of the population is selected. So look at this one. In the first graph the selection is from both the side, in the second graph selection is on one side, and in the third graph there are selection of the population from the middle one. So look at this one. If that is the selection, and selection is in the from the both the corners. Now if that species is not selected, this one is not selected, this one is not selected, so we have to name these one. So first question of this one is, what names are given to the type of selection? What is the type of selection in three graphs, in the first one, in the second one, and in the third one? And second question is, after the selection has operated, now selection took place and natural selection took place, then after, what is the result of that natural selection? So look at this one. So all the graph, so if we are not selecting from side one, that is the stabilizing. If we are selecting the not selecting the species from one direction on one place, and rest of the species are selected, that is the directional one. And disruptive means if we are not selecting the species from the middle one, and we are natural selection is acting on the species on the both the sides, that is known as disruptive. So because of this, what is the conclusion? That's second question. And in the second question, what is the conclusion? That graph first is stabilizing, graph second is directional, graph third C is disruptive. That means after the natural selection is acting on these one, what is the conclusion? So conclusion is this one. Look at this one. In the first selection we have not selected the species from both the sides, so that is the conclusion of the natural selection. In the second one we are not selecting species from the one direction, so that graph B is directional, so that is the one. And in the third one, that is the, we are not selecting the species from the middle one, so because of that reason there is a disruptive graph for the natural selection. So these are the three graphs after the natural selection acting on this. Now next one is the aeration tank. There are the sewage treatment plants, and in these sewage treatment plants they are acting, and because look at this one, if there is a respiration, that means they are consuming oxygen, they are mostly consuming oxygen, that means aerobic respiration, and that for particular oxidation, particular consumption, particular degradation of that particular organic compound, there is a requirement of oxygen. And what is that particular requirement? That is known as BOD, biological oxygen demand, or now it is known as biochemical oxygen demand. So if in a treatment plant that is not working properly, so what is the effect of these phenomenon on the particular microorganisms which are growing in this one, and they are degrading the organic material? So look at this one. It will adversely affect the secondary treatment or biological treatment of sewage. If biological treatment is taking place, that means they are using the biochemical reactions, and they are using biochemical reactions, that means they are utilizing the oxygen, and when they are utilizing the oxygen, then aerobic respiration is in disorder in that particular sewage treatment plant. When the aeration tank is not functional, the air will not be able to pump in this one. And what is the effect? When the air is not able to enter, that means they are not able to carry the oxygen into the treatment plant. So look at this. This will not allow the vigorous growth of useful aerobic microbes into flocks. That means these microbes are not able to grow with that much speed, and when they are not able to grow that much speed, they are not able to degrade that organic matter in this one. So look at, thus the major part of the organic matter is in the effluent will not be consumed by this bacteria. And because of this reason, biochemical or biological oxygen demand of effluent will not be reduced. That is keep on increasing because the oxygen is not available to that particular bacteria to act on these organic food. So because of this reason, BOD refers to the amount, as we have already discussed it, BOD refers to the amount of oxygen that would be consumed if all the organic matter is in 1 liter of the water were oxidized by bacteria. So that is the particular 1 liter is given to measure that biochemical oxygen demand. Greater the BOD of wastewater, more that is polluted. So because of that reason, sewage treatment plant must be acting properly. Thus the effluent will remain polluted with the amount of organic matter and high BOD. So that is the effect of non-functionality of the pumping or that sewage treatment plant. Next question is, again we are going to the biotechnology. A farmer grow 2 varieties of corn crop in a field A and B. He grows normal corn crop in field A, genetically modified corn crops in field B. He observed corn borers attack. That means there are species of BT cotton were spread in the BT cotton virus spores are spread in the field A. So that means there are 2 fields. First A field is they are growing that plant, corn plant. If that plant is growing in that field, they are normal crop. In the second B field, they are growing the genetically modified corn. But what is the effect is that in the first one they are spraying that BT spores into the A field. So what is the effect of this one? So the first one is name the gene in the spore responsible for control of this pest. So we have to name that particular gene, which is the name of that particular gene, which is very much useful to control that particular pest. Second question, what effect will be spores of BT have on the insect pest? That insect pest, what is the effect of that gene on the insect pest? Third one is, how has field B developed resistance against this pest? So we will go to this one. So first one is, that is the IAB gene is the name of the gene which is responsible for that BT, Bacillus thuringiensis, that gene which is the pest resistance, and we have developed the BT cotton also in this one. The spore BT cotton, that means that BT cotton is in the spore, that is in the inactive form, and when that is converted into the active form, that means that is acting on a particular pH. So if pH is alkaline, that means that particular protein toxin is converted into an active form. So whenever that protein is inserted, that toxin is inserted into a insect gut, that means they found the alkaline pH, and in that alkaline pH they are able to convert it into active form. And when they are able to convert it into active form, now if it is converted into active form, that means that affect the gut of that insect, and when that affect the gut of that insect, that means there is a abnormal situation, and that abnormal situation is become porous, that gut is become porous or swollen. Okay, so because of that reason, that insect is quickly dying. So because of that reason, that is the effect of those spores of BT. We are infusing that one in the field of A. Now second one is last one is that Bt gene is cloned, inserted into the plant genome. That is the biotechnology, and in that biotechnology, that is very much useful to Bt toxin, and Bt toxin is Bt cotton. That is very much useful crop to pest for the pest resistance. Now next one is give the 3 reasons to why prokaryotes are not given any figure for their diversity by the ecologist. Look at, there are huge diversity of the bacteria, there are huge diversity of prokaryotes in nature. We are not able to, we are keep on developing our new technologies, so we are able to identify these huge variety of bacteria in the nature. So because of that reason, whatever be the technological advancement that is coming, by these technological advancement we are able to identify. So now we are not able to identify that much diversity of the prokaryotes or bacteria by recent or the present technologies. So because of this reason we are not able to give the number of in the diversity. Prokaryotes organism diversity is not given any figure by ecology because of the following reasons. So that is a classification and identification of vast majority is not, we are not able to now we are not able to determine that much of. Now second one is, for many microorganisms it is difficult to culture them. Yes, that is now whatever be the cultural techniques we are having right now, by these cultural techniques we are not able to identify these one, we are not able to culture these bacteria. So because of that is the second reason. Third reason, according to current biochemical and molecular techniques, it is estimated that microbe diversity can range in billions. Look at the diversity. So because of that diversity reasons, we are the scientist is not able. So hence more advanced molecular and biotechnological techniques are needed to classify, to identify, to characterize, or to name, to give a particular number of that prokaryotic organism. So because of that reason we are not able to give the particular number of the diversity of these organisms. Now that is the completion of section C. Now section D. In section D, the structure below, so there is a plasmid, and this plasmid, this one, which is similar to, there are two plasmid, PUC18, PBR 322. There are two plasmids, and that is the structure of plasmids. So how these two plasmids are different from each other, and why that, which one is very much useful. So look at it. Now, there are plasmids, there are two types of these plasmids. First one is that these are natural plasmids, and these natural plasmids are present in the bacterial cell. So they have the particular sequence, they have the particular genetic material, they have the particular genes, they are useful for the bacteria. But we are utilizing these plasmids in our biotechnological experiments. If we are utilizing these plasmids in biotechnological technological techniques, that means we have to modify these plasmids, those which are naturally present. So we have developed some artificial plasmids, and these artificial plasmids developed according to our human requirement, and by these requirements we are able to fulfill these technological advancements. So in this one, because look at it, if there is a plasmid is working, that means that plasmid is able to replicate, that plasmid able to incorporate, that plasmid able to identified by the human being, okay, this one gene is incorporated or not. So we are inserting all these features into a plasmid. So these are artificial plasmids we are developing, and in these artificial plasmids we are developing those genes, we are incorporating those genes which are useful for us in the biotechnological experiment. So first question, how are PUC18 and PBR 322 used in biotechnological studies, that is studies in the form of vector? Now second thing, look at, we are using biotechnology. In biotechnology, recombinant DNA technology, if we are inserting one DNA into another one, then we have to cut the DNA. Again we go back to the bacterial cell, and when bacterial cell we have identified a very much important enzyme, and that enzyme is restriction enzyme. By that restriction enzyme we are able to cut DNA a particular place, and that particular place is known as restriction site. Now if that restriction site is present, that means that restriction site is must in a particular sequence. That sequence is known as palindromic sequence. So again goes to the bacteria, and bacteria provide us a very much important enzyme, that is restriction endonuclease. Restriction endonuclease can cut a DNA at a particular place. That particular place is known as restriction site, and that restriction site is in palindromic sequence. So there is a sequence. If like Z gene recognition site is there is a many genes, there are many sequences present in a particular gene. So what is the sequence at which place that particular enzyme will cut? We have to identify that particular sequence in this one. Now third one is applying your palindromic sequence. How that palindromic sequence is acting. What is that particular sequence that acting as a palindromic sequence, which is the place in this one they are cutting the DNA. So look at this one. There is a vector. In that vector there are sequences. In these particular sequences we have to identify that particular restriction site. We have to identify the palindromic sequence. And now next one is restriction enzyme. Why you used to extract the interest of the plant. This gene needs to be inserted into DNA segment which has been treated with the restriction enzyme. Why? Will there be a successful recombination? Explain with the reason. How we have to identify that particular this one is the gene we have inserted into a particular place. And last one is C. Which one of the two, PUC 18 and PBR 322, would be prefer for your biotechnological studies, and why? So first one is plasmid, and plasmid are using as a vector. So look at this one, plasmid, which can be used to insert the gene of interest from a desired organism into a host, or they act as a vector to transfer gene of interest into the host. So we are using a vector to carry that gene to a particular cell. Now second one is that this one is the our B1, and look at this one. They are cutting the DNA. This one is the restriction site. This one is the restriction. So they are flanking regions. These are the flanking regions which are acting as anchorage sites. Okay, so this one is the second. Third one is second part of the second. No. Is the restriction enzyme needed to be same which cut the DNA of plasmid and gene of interest from the plant? Look at this one. On one side there is a vector. On the another side we have that particular gene we have obtained from a particular place of the DNA. Now if we are able to insert, we are wanted to transfer that particular gene into a vector. That means we have to cut that plasmid and that the segment of gene from one restriction enzyme. We are not able to use two different restriction enzymes. We have to cut both plasmid and that gene from a same restriction enzyme. So we are not able to separate that. We are not able to use two different restriction enzyme for the same segment of plasmid and that DNA. So next one is, because PUC is more that have the more character, that artificial plasmid has the more character. So because of high copy rate of this one, what is copy rate? That means they are able to multiply. They are able to multiply very much frequently. So that is known as copy rate. And because of PUC 18 has a more copy rate, so that is because of reason we are able to utilize PUC 18 more quickly than the PBR 322. Now next one is there is a two graphs, and in that two graphs there are two organisms are growing. In that two organisms are growing, that means population first and population second. In this one, look at this one. This one population size, this one is time. With the time, these populations are growing together. On the other side, this population is stagnant, and the second population is exhibiting, the B population is growing. So they are asking that graph A and B shown below depict interaction of two species, which graph indicates mutualism. So if there are mutualism, that means there is a ecological hierarchy, there is a species converted into population converted into community, and community lastly converted into a ecosystem. So at the community level, the different populations are growing on a particular place. When the different populations are growing on a particular place, that means that population, there is a interaction of population at that place, because different populations are growing together, and because of that reason we are studying one topic. There is interaction of population, and in that interaction of population there is the different combination, there is different relationship between the different populations. So out of these relationship, one relationship is mutualism, and in that mutualism means both the species are growing together and they are benefited together. If they are benefited together, that means they have to grow in a similar way. So which graph is showing the exhibit mutualism? So graph first one is showing mutualism, because these two populations are associated with each other, and they are associated in such a way that relationship is beneficial for both the species. So because of that reason they are able to grow in a similar way. So graph A is the exhibiting the mutualism, and because they grow simultaneously. So that is the completion of section. Now last section is section E, and in that section the first question is, given below are certain situations, analyze the situation and suggest the name of suitable contraceptive devices along with the mode of action. That is the reproductive health, and in that reproductive health we know that there are population control, and in that population control we are utilizing some mechanisms by which we are able to control the growth of the population, and there are research, and in that research we are able to develop some technologies. So first one is that blocking the entry of sperm through cervix. We have to stop the entry of that sperm into a female body. That is the first one. Which technique we have to utilize? Second one is spacing between the two children. We would like to create a space between age difference between two children. So what is the gap? How we have to develop the gap between the two children? So for that one we have to develop that technique. What is that technique? Third one is effective emergency contraceptive. Which are the we are mainly utilize contraceptive. Fourth one is terminal method to prevent and more pregnancy in female. To stop the pregnancy in female, what type of techniques we are utilizing? Sterilization in male. What is the name of that technique? And now first one is in the answer. For the blocking, for the entering of sperm into a women body, we have to develop diaphragms or cervical caps or vaults to stop the entering of the sperm into a body. Second one is spacing between the two children, because we have to create a gap between the first child and second child. So there are intrauterine devices. There are many devices they are utilizing. So look at in these devices they are mainly utilizing copper, and that copper is that made the viscous. That fluid is made the viscous because of that reason, and that copper is also for the for very much useful for the women. Because of that reason, they are mainly developing the intrauterine devices, and these devices contain that copper. So copper hormone releasing in IUD such as copper T है, copper 7 है, और multi load 375, progesterone LNG 20. There are different the techniques. They are mainly utilizing the copper. So these are the usefulness for the women body also, and how they are acting these techniques. Now third one is effective emergency contraceptive. That means there is a relationship, and in after that relationship, how to stop that particular sperm into the women body to become or to fertilize the egg. So these are the pills. These are the mainly pills, and these pills are mainly utilized to stop the entered sperm to fertilize with the zygote. Now next one is terminal method to prevent any more pregnancy in female. So that is our permanent solution. We are utilizing this technology, tubectomy. We have to cut that tubes, fallopian tubes of the women. So because of this one, block the gametes entry of that transport and prevent conception. The fifth one is sterilization in male, and that is sterilization is vast differences, and these tubes we are able to cut these tubes. That is vasectomy. Now the next question is 32nd. That is from genetics, and in that genetics that there is a cattle, and that cattle has a two color. That is the coat color, that is a red color, and that is the white fur, and we are interbreeding these one. When we are interbreeding these one, after that we are developing that next generation, that progeny from these one. So what is the effect of this one? What is that condition? Is that, first question, is this an example of codominance or the incomplete dominance? Second one is give a reason for your answer. Third one is with the help of genetic cross explain what will be the consequent phenotypic of calf when red is dominant over white. So first one is that red is dominant and white is recessive. Second one is red is incomplete dominance. So what is the answer? That is the example of codominance. Codominance means the expression is of both the genes, that is the recessive gene and the dominant gene, and because of that reason there is a expression of both the character, that is dominant character and the recessive character, that is known as codominance. And because of that codominance, both the characters are expressed. Codominance is a condition in which two different alleles for a genetic trait are expressed. That means they are, one is dominant and one is recessive. But in normal condition, we are saying that dominant is expressed and recessive is not expressed. But in this condition, both are expressed. Now, how that breeding behavior? The first one is that if pure breeding red coat cattle are represented as R, and pure breeding white coat is small r. So that is expression, and in that expression red is dominant over white. What is the effect? That is the effect. In that effect, the phenotypic ratio is 3:1. How that phenotypic ratio is 3:1? Look at this one. First one is red, that is the dominant one, that is the second parent, or that second parent is recessive, that is a white. So there is a gamete formation. In that gamete formation, that all the gametes contain the R gene, all the gametes contain this gene. Now after that one, the progeny, the F1 generation, and that F1 generation is from they have the gametes, and these are the gametes. Now in this one we calculated the phenotype, and first condition where the red is dominant over the white, in that condition the phenotypic ratio is 3:1. 3 is red and 1 is white. Now next one is in the form of codominance, where both the genes are expressed. So when there is a codominance and in the form of a incomplete dominance, at that time both the genes, the dominant gene is not able to express. When the dominant gene is not able to express, that phenotypic ratio will be changed. So if this one is the phenotype of an organ or genotype of an parents, then the generation of that incomplete dominance, the phenotype is 1:2:1. How that happens? Look at this one. This one is the parent in the form of incomplete dominance. That means dominant is not able to express that that particular feature in a dominant form. So look at this one, that is a gamete formation, capital R, small r, and after this one, that is the homozygous dominant. So that is exhibit the red color, that is the heterozygous condition, and when they are heterozygous condition, they exhibit the different color from the red and white. So that is the pink color. Now in the third one is, that is the recessive condition, and because of that recessive condition, they exhibit the white feature. Okay, so phenotype is in this condition, phenotype and genotype will be same. So the phenotype is red, pink and white. Now in the last question, and that last question is, with the help of a flow chart, illustrate how an infected animal cell can survive while viruses are being replicated or released. So we know this one, virus are made up of only RNA or DNA and a protein coat. So there are some DNA viruses, some are RNA viruses. They are inactive outside. When they enter into a living cell, then they are acting as a living structure, and because of that reason, how they behave. So in this one, there are two type of cycles. One is lytic cycle, lysis cycle, or second one is lysogenic cycle. In lytic cycle, they destroy the host cell. In lysogenic cycle, they are completing their life cycle according to first complete that life cycle, then release from this. They are not destroying the host cell. So in this one, if we calculate a virus which are able to enter into a animal cell, and when they are animal cell, that means they are mostly containing RNA virus. So these viruses are known as retrovirus, and in these retrovirus, genetic material is made up of RNA. Now that is a protein coat. When that infect the host cell, animal cell, when they enter into animal cell, the coat is remain outside, and only the DNA is able to enter into the host cell. And now this one is the RNA is a genetic material. Now they utilize reverse transcriptase enzyme, and that reverse transcriptase enzyme is able to convert this viral RNA into a DNA. So there is a reverse transcription, and now this is converted into DNA. That DNA is able to incorporate into the host cell's DNA. Now this will control the host cell's mechanism, all the mechanism. When that is able to control the host cell mechanism, then that is able to replicate. Now it will replicate and form its own copies. When that is formed own copies, a fixed number of copies is formed, then that protein is synthesized. These proteins will cover the host cell, and when they cover the host, cover their DNA, when they cover their this genetic material, they are converted into a new particle, that virus particle, and these virus particles are released from this one. So that is a life cycle of a virus into a animal cell. So today we have discussed the one model paper for this CBSE 12th. Now you have to solve according these objective questions and write the answer. These answers you have to find out another questions similar to this one, similar to these topics. Okay, this one is the topic and this one is the maybe question is framed in this way. So you have to develop some techniques to analyze topics around these questions, and by these questions you are able to take a control on your subject. Okay, thank you very much.